Good morning, everyone. I'm Tony Stokey, a postdoc from Pennsylvania State University. And today we're going to be talking about EDNA. It's a program to use metagenomic data in a diagnostic sense. So just a quick introduction. So next generation sequencing is not a new technology anymore, but when we started this project it was. It's a way of getting hundreds of thousands of different sequencing reads. There are many different platforms, each with their own particular flavor. Roche 454, which isn't really supported too much anymore, Ion Torrent, Illumina, Pacific Biosciences, all of them have their own uses and different, different flavors. The basic way that the chemistry behind it works is you amplify a single nucleic acid molecule on a bead or a polyne, and then visualize the incorporation of bases either through pyrosequencing, measuring pH in various ways. But that visualization then leads to a large number of sequences. With this new technology, we've really looked into this field of metagenomics, where you combine the genetic material of a given environment. This is really useful because you can sequence organisms which are hard to culture or just simply cannot be cultured. You can also look at the interactions between these organisms and look at the gene expression of a given environment instead of under ideal lab conditions. But what we're really interested in is using metagenomics as a diagnostic tool, looking for specific pathogens of interest. Why should we do this? Mainly because viruses and pathogens in general. I'm a virologist, so viruses are my pathogen of interest. Viruses are moving all around. Some of these numbers are a little bit old, but in 2012, 62 million metric tons of foodstuffs have been imported into the States. It just leads to a large number of ways that a virus or another pathogen can enter into the United States. This is important because agricultural exports and imports adds $21 billion to our GDP, and almost 2/3 of losses in crops are due to these exotic pathogens. So it is very important to screen imported materials coming into the States. So next generation sequencing can be used for this. Instead of looking at a given environment, we can look at a plant as the environment. And so what's inside the plant is the plant host, the endophytic bacteria, fungal sequences, and also any pathogens which are present. And we're only really interested in the pathogens. There are some problems with using...
Okay, so how do we hit the speaker?
I'm sorry?
We got it, Dr. Dennis. I can hear them. So how do we hit the speaker?
Okay. So there's some problems with using next generation sequencing as a diagnostic tool. In general, just this idea of big data. It's a large problem, because it can be very time intensive, computationally challenging to shift through all of this sequencing data. Okay, so next generation sequencing, the problem, big data. Big data can be very computationally intensive to search through a large amount of sequencing data, especially when you're only really interested in a very small portion of that. Not only that, but there's also problems with just storage. Where do you store all of this data? I saw a talk recently of someone doing cancer sequencing, and his unit was how many Large Hadron Collider projects does it take to hold this amount of data. It's just going to be a big problem coming up. So this is kind of our workaround. Our solution to that is an eProbe diagnostic nucleic acid assay, or just EDNA, because we need funny names. So EDNA, what exactly is EDNA? It is a way of using a set of known pathogen-specific sequences. So these are curated sequences, which we're calling E-probes. You can think of this as kind of an analogy to microarray probes or specific PCR primers. So these are designed sequences that we use to query against raw sequence data set. So this is a sequencing data set that hasn't been filtered, hasn't been QC'd or assembled. This is just the raw file. So the E-probe design is a very important part of this, of course, because if you have bad PCR primers, bad microarray, your test is not going to be very accurate. So the E-probe design is somewhat straightforward. You compare a target genome of your pathogen of interest with that of a near neighbor. So if you're interested in plumppox virus, you want to find another potivirus. Compare the two and remove anything that is similar. So you're left with the target unique sequences. These unique sequences then are, you remove any homooligomers. We were using 454 sequencing, and homooligomers are a problem with that, so we removed that. But you also do a blast check to make sure these pathogen unique sequences aren't hitting on your host or things that are common, zebrafish or whatever. So once you have, let me go back. Once these are done, you also have a set of decoys, which are basically these E-probes in reverse. Not reverse complement, but the reverse. So it has zero sequence similarity, but similar GC content and things of that nature. So once you have your E-probe design down, you have your target and your decoy E-probe sets. You have your simple sample database, your sequencing data set. You then blast your target and decoys against the sequencing data set. You score them, and then you check to see if the target scores and the decoy scores are similar. If they are similar, we can say that the target or the pathogen is absent. If the target scores are significantly higher, then you can call the target being present, or the pathogen is present. So how do you go about scoring these E-probes? Like I said, in BLAST, I'm sure everyone's familiar with BLAST. This is a BLAST output. Our scoring system is the negative log of the E-value times the number of identities times the length, or over the length of the probe itself. So in this situation, you have an E-value of 0.17. Length of the E-probe is 64, and the number of identities is 15. So in general, this is not a very good hit. This would, if you looked at a blast of this, you would say that this isn't significant in the least. And that, our score then is 0.18. On the opposite end of that, if you have a E-value of 10 to the negative 21st, 63 out of 75 of the E-probe is identical. This is a very high score, which is what's shown here, a score of 10.62. So you have, and just like in BLAST, you have multiple hits for a given query. With EDNA, you can change the number of top hits you want to look at. So if you have a very low titer virus, you may only want to look at the very top hit. If you have a very high titer virus, you want to look at all 10. So then we combine all of these different hits in our scoring system. So like I said before, we are comparing the target scores versus the decoy scores. And to do that, we use two different ways. The first one is just a simple t-test to compare the two. The other thing we can do is look at individual probes by taking the average of all of the decoys and then comparing the target score of an individual probe to the average of the decoys. So this is kind of like the baseline noise that BLAST is giving, and then each individual target is to check to see if it's significantly higher than that average. And in silico testing, just to show you that yes, it does work. These are all p-values. We are able to detect down to less than 0.5% or 0.5 to 5% of a metagenome sample of the reads being pathogen. We're able to detect it fairly consistently. And we can detect down to less than 0.5% of pathogen reads in a metagenomic sample. Okay. And with that, I'm going to pass it along now to John at Oklahoma State.